So first of all, thank you very much to Thermo Fisher and Kevin for giving me the opportunity to tell you about the external quality assessment story in this field. So, as Kevin says, hopefully the majority of you in the room will know about UK NEQAS in some shape or form. But for those of you who either don't or won't admit to it, I've just got one slide to summarise what we are about. So UK NEQAS is the UK National External Quality Assessment Services. And really what we're there to do is to help ensure that your clinical lab test result is accurate, it's reliable and comparable wherever it's produced. So if someone is tested in the UK, they get exactly the same result than if they were tested somewhere else, for example, in the States or across Europe. UK NEQAS itself is a charity organisation. It's an umbrella organisation, and there's a central office in Sheffield in the UK. And I think it's very important for participants to know we're self-funding, non-profit making schemes. So even though we do charge a participation fee, any funds that are left over are rolled over to develop new pilot schemes and to respond to participant needs. And this NIPT for Aneuploidies pilot scheme is an example of that. There's over 25 specialist centres currently dotted across the UK, so we're not in one central location as people often think we are. And really that's historical. We've grown up from specialist interest in expert areas where people have an interest in quality in their field. And we cover the majority of all of pathology medicine now, so there's schemes from biochemistry schemes, which you probably know and love so well, right through to haematology, microbiology, and then the latest scheme, which is the one that I run, is molecular genetics, but saying that we've been around for about 25 years now. Moving on to years, over 45 years experience providing EQA within the NEQAS organisation. So I'd just like to say I've not been around for 45 years in NEQAS. And as well as the labs under pressure now to implement and adhere to ISO standards for their accreditation, all of the NEQAS schemes are ISO 17043 accredited schemes as well. Now, despite being called UK NEQAS, we actually have participants worldwide, and we're just based in the UK. And my EQA scheme itself has covered 68 different countries during 2016. And I would say about 70%, in particularly in the specific testing world, as in aneuploidies and also pre-implantation genetic diagnosis, about 60 to 70% of our labs are based out with the UK. And just to give you an idea of how molecular testing is starting to invade all types of laboratory medicine, you can see here up till about 2006-07, we only provided about 10, less than 10 EQA schemes, and you can see how that's grown considerably, and we now are over 50 for 2017. So a very quick summary of the principles of EQA, because as I go on to develop how we've expanded the NIPT pilot, then this will help you understand some of the problems that we've had. The main principle of EQA is you can send the same sample to all the participating laboratories, so you actually can make some conclusions. You can look at different methodologies, you can look at different DNA extraction methods, you can look at different strategies of testing, and we can do that because you can benchmark, because everyone is receiving the same sample from the outset. The molecular EQAs are slightly different from some of the other EQA schemes that you may participate in such a way that they're not qualitative. A genetic test result tends to either be the abnormality is present or it's absent. We're not looking for values, although we can go into the realms of fetal fraction, etc., which we'll touch on later. So what we ask for is for participants to interpret the result in the context of a clinical case that we supply with that sample. So we're assessing for genotyping accuracy. So basically, have you got the right result? Is it correct interpretation of that result? And often we see laboratories who perhaps are not quite up to date with the current literature and actually are giving incorrect or misinterpreting some interpretation that the reader of the report should take into account. And we also look at the clerical accuracy of the report, because you would be absolute, or perhaps not, be absolutely amazed how many patient names, dates of birth, sample reference numbers, etc. are incorrect on a report. And if you're reporting a molecular test result, then how can the reader of the report be sure that that is actually the right result when you can't get the patient details correct? So even though we don't base performance monitoring on clerical accuracy, we always do report back to the labs. Now, it's not just me sitting in the lab in Edinburgh deciding who's going to get good EQA scores and who's not. We have agreed marking criteria, which is ratified by peer specialist advisory group. And for the NIPT Aneuploidies schemes, we have an advisory group set up for rapid prenatal diagnosis with a lot of expertise, both based in the laboratory and clinical input. All of the reports are marked independently by a team of assessors, and they are all anonymised. So even though you may suspect you recognise that report from your colleague down in at the other end of the country, there's no way that you actually confirmed that you know who wrote that report. Also, because we have assessments throughout the year, this means we've got continual performance monitoring, and also one of the aspects that ISO and our other accreditation bodies like is the fact that labs have ongoing records of participation and performance. So it's all stored online, so labs have always got access to their own data. So in summary, EQA provides labs with an external measurement of their standard of service, and along here at the bottom you can see just some examples, and there are many, many others, of the different bodies that actually use EQA data to base decisions on. Albeit clinical users, will your local clinicians or external clinicians use your service? Subcontracting laboratories, if you're outsourcing, you want to know that they're actually a good lab that you're sending your samples to. Commissioners for funding, research and development partners, for example, performing testing of patients perhaps being included into clinical trials here. And also one of the main issues that I think is just fantastic about EQA, and it may just be a personal thing, but it's about the amount of data that we can collect. We can find out who is doing what, where, and how they're approaching it, and get very first indications of when things maybe not be optimal, or when an assay or technique comes in to practice that is actually excelling, and we can feed that information back to our participants. And I think because we are independent, we're not related to any company or funding stream, then we actually have some value with the data that we have. Okay, so before we move on to NIPT in particular, I just wanted to give you a flavour of how molecular testing is now extending, as I say, into laboratory medicine. So we started off up at the top with our yellow circle of monogenic diseases. Now this was our bread and butter. This is where we started from. This is your cystic fibrosis testing, your BRCA1, BRCA2, Huntington's disease, etc. But we no longer test just solely for monogenic disorders. The genetics laboratories are turning into genomics laboratories. They're combining with cytogenetics laboratories and molecular pathology labs. So we also had to extend our scope of EQA. For many years, we have been providing newborn screening EQAs, and that really is dependent on the country that the laboratory is situated in. But in the UK, we are asked to provide by the screening committee, the National Screening Committee, to provide the EQA for the reflex molecular testing for cystic fibrosis and for MCAD. So we send out Guthrie card, blood spot cards to mimic the samples that the labs routinely test. There was a question briefly earlier about PGD. We also provide PGD both for monogenic disorders, also for aneuploidies and translocations. So again, that touches onto NGS and arrays, and many patients undergoing IVF, again, country dependent, will request for screening for trisomies and aneuploidies and translocations prior to implantation of the embryo. We'll come back this way. Molecular pathology is something I don't know how many of you are involved in, but this is something that has become expanded dramatically over the last few years in the field of lab medicine, in particular for companion diagnostics and personalised medicine. So we're heavily involved in molecular pathology. Next generation sequencing pilot. This is one of our EQAs that's actually a technical-based EQA, and that's because NGS is being used in a huge range of applications now. So this is a pilot scheme that you can participate. Doesn't matter what you're testing, how you're testing, doesn't matter which platform you're using. You get a measure of your accuracy of your variant calling and the quality of your NGS data. We're also involved in the 100,000 Genomes Project in the UK, again, because genomic medicine is here to stay. And we are looking at individual competency and trainings and tumour assessment EQA schemes online. But we're here today to talk about the NIPT testing pilot, which has been a long and sometimes difficult story. Thanks to the two presenters who kicked off this afternoon session, I don't have to do any introduction about NIPT because it was so very well covered. And this is just one example of the testing where it's actually looking at the quantity of reads for different areas on your chromosomes, hence why you can pick up whether there's aneuploidy present or not. So it really started through participant demand. So as I mentioned earlier, we're here to deliver EQAs that's going to benefit you, because that's what we want to do. And I think over the last few years, there's been a shift to the people's views on EQA. It used to be something you always had to do. Now people are looking to us when new tests are coming in and are being driven to be delivered in the clinical field to actually have some sort of assurance yourself that you're doing, one, the right thing, the same as everyone else, perhaps struggling with the same issues as everyone else. And so really this participant demand was quite strong. It's technically challenging to be able to source samples that really reflect patient material. The last thing we want to do is send out EQA samples and actually assign performance scores on those if they're not actually fit for purpose and labs can't perform their routine test. That wouldn't help anyone. Logistically, it's challenging. Sending plasma with very small fragments of DNA that's terribly unstable across the globe is no easy matter unless you have millions of pounds that you can send everything on dry ice. Dry ice costs about 500 UK pounds per shipment, so not feasible really. Difficult to obtain your patient material. There's ethical issues, availability. How many people in the labs have a lot sample that they can store? We couldn't send the same sample to absolutely everyone. There would be one poor, very pregnant lady who had a very sore arm. It just wouldn't work. And also, if we're looking at some of the rarer trisomies or aneuploidy or monogenic disorders, there's disease incidents to think about as well. If it's very rare, then obviously we can't just pick something off the shelf to use when we wish. So really, the demand was much bigger than one EQA provider. So UK NEQAS for molecular genetics took the lead, and this was because previously we have been collaborating with EMQN, which is a European network based in the UK, on sex determination in plasma samples. So we'd put a lot of research and development work into working out how we can deliver plasma and the logistics behind it all for sex determination. But then that's much easier because you can pool your samples if people are using particular technologies, which you cannot do for aneuploidy testing. We also go back a good 10, 12 years with our cytogenetics EQA peers, who we have run the molecular rapid aneuploidy EQA scheme. So this is your routine 24-48-hour turnaround test for your trisomy 13, 18 and 21, albeit QF-PCR, MLPA, BOBS, whatever technology. So again, it shows how genomic medicine is integrating and coming together, and it doesn't matter what lab you're working in, it's the expertise across the field that we need to deliver this. So the first thing we needed to know was what the labs are actually doing. What do they test? How do they test? How much do they test? When do they test? So we sent out a laboratory survey at the beginning of last year. It went to over 1,200 different laboratories across the globe, and we wanted to know about NIPT for aneuploidies and for sexing, and any plans or current services for single gene disorders. We had a response from 121 laboratories, which sounds low, but considering after seeing the scope of our schemes, you can see that many labs don't actually do this specialised testing. And 61% of the labs currently provided NIPT for aneuploidies. We also asked, if you don't currently provide it, are you planning to set it up as a service, and when do you expect that to be in place? And all of the rest of 59% of labs were going to set it up and plan to have it in place by the end of 2016. So you can see now the data, if we did the survey now, would probably be very different with everybody providing NIPT for aneuploidies. There was a huge range of the number of samples tested per month by labs. We had down to 5, 10 per month, right up to the 1,000 benchmark. So the mean turned out at 565 samples. So I think the message from today is this test is here, it's being performed, and I guess we need to provide our EQA to make sure it is happening well. The next few pieces of data show you really the variation of approaches and how people are starting to perform this test. Here, this is the volume of blood required. So you have some labs that just saying 4 mils, right up to 20 mils, poor lady, but with an average about 10 mils. So this is how we can start feeding this information back to the participants, and they can say, well, what are these labs doing differently to these here, and maybe you can optimize your testing in some way. Turnaround time ranged from 0 to 3 days, 4 to 7, 8 to 14 is the most common, 15 to 21 days. But good turnaround time doesn't always necessarily mean accurate, good testing. Not only the blood, but we also wanted to know how much plasma labs required for testing, and often this was dependent on whether they repeat the testing, run it in parallel, or store plasma for testing. And this was also information that we needed to know how much we needed to supply labs to actually perform the EQA. And again, the average is about 3 to 4 mils, but some people want up to 10 mils of plasma. I guess it's this 20 mil blood lab, but who knows. A lot of the issues on this slide have been touched earlier today, and we did ask a quite extensive amount of questions. We wanted to know, did you test for sex chromosome abnormalities? Did you test the fetal sex? And many assays do test the fetal sex because they require that for interpretation of the result. So we also asked, do you report the fetal sex? Because that's two different things. Do you measure the fetal fraction as part of the assay? And again, do you report the fetal fraction as part of the assay? Also, what information is required for testing? And we gave a drop-down list, and it was very much what's been mentioned this morning, this afternoon: IVF, twins, BMI, screening results, anything, maternal age, gestational age at time of sampling. So we had a big list of things, and it was actually quite enlightening to find out which lab, that many labs didn't actually ask or request for the extent of the information that you would assume they would need to interpret the result. So there was obviously a learning curve there. Some people just would take the test and would take the sample and just perform the test. Many labs actually outsource their testing. So we had a stream of questions about, if you outsource your testing, do you want your outsourcing lab to participate in EQA? Would you send them samples to check? Would you ask for the results to come back to you? And again, it's taken that responsibility for the level of service despite where it's performed. We also asked for future plans for testing for other abnormalities, because we know this is happening within the PGD world with screening and full genome arrays and NGS, so we wanted to know if this was coming into the NIPT or NIPD world, and also future plans for NIPD, because this is the next project that I'm going to take on this year for my sins, I think. Thought you would just like to know, everybody likes to know what tests other people are performing, so this is just a snapshot of the methodologies that people are performing, and no surprise, the most common is next generation sequencing, but there was a handful of labs doing Sanger sequencing. I'm not going to comment on any of these. Sanger sequencing, arrays, MLPA, TACMAN, dosage assays, and some people didn't know what they were doing, which is a bit worrying. So We see that a lot, don't joke. So the next stage, now that we sort of scoped out what was happening, the next stage was how do labs actually report the results? So we thought we would embark on a study about reviewing the reporting styles. So we asked we asked all the labs that had completed the survey, but 45 labs came forward to say they were very happy to submit us anonymised, a low-risk aneuploidy report and a high-risk aneuploidy report. And we went on to hold a panel discussion, very interactive, I would say, panel discussion at ISPD 2016 workshop pre-congress last year, which was actually in Berlin in July. I'm not going to talk about the results because we have just submitted, and actually it's now in press, a manuscript which is the recommended practice for laboratory reporting of non-invasive prenatal testing for trisomies 13, 18 and 21. Now this is not a best practice guidance document because there is not a clear steer on many of the issues. It's purely a consensus opinion. So we went through all the findings of the content of reports, and if an agreement was made that that was absolutely essential to be on your report, for example, things like maternal age at gestation, other screening data, for example, then that has gone in as a recommendation. Anything that was very heatedly debated, and we often had to have a vote, and there was not a consensus opinion, has gone into the manuscript as no agreement was made. For example, fetal fraction being stated on the report. There was 2 extreme views in the room, and nobody would budge. So I guess it's something that is very to be debated in length. Stage 3 was more about providing the samples and how we're actually going to run the EQA. How are we going to do this? So we sourced 3 different types of samples. We used very kindly the patient plasma samples stored in the UK Rapid project, because we all know that you can never get away from the fact that a real patient sample is going to be the most optimal for EQA purposes. We also know that there's artificial reference material kicking out about there. There are many companies trying to produce artificial material to get into the market to help labs with their validation and verification of assays. So we wanted to make sure that, we've been working closely with them, but we wanted to make sure that we could say to labs, either, yeah, these are fit for purpose, or I would maybe hold off till more work's been done. So I'll explain shortly what A and B means. EQA schemes have to be method agnostic, so we want it to be available to anyone who's doing the testing. So for this exploratory pilot, we used whole genome sequencing with Illumina with different protocols. So 3 labs were using Illumina platform, but different protocols. We were using the Life Science Thermo Fisher Ion Proton NGS method, and also SNP arrays. So we were, at that point in time, we were covering pretty much everything that was happening. And we included samples trisomy 13, 18, 21, and Aneuploidy for testing. So this is a summary of our patients, the plasma samples. So we covered all 4 options. These labs here are the Illumina NGS labs, and you can see here that everything in green concurs with the results that we expected. The Ion Proton NGS panel also concurred, and the SNP array. So we were hunky-dory, great, really happy, but then we knew the patient plasma should be working. But it did endorse the fact that we sent these plasma samples out at room temperature, ambient temperature. They were not on dry ice, they were not on ice, they did not go out frozen or refrigerated, and they were global. So we do know that how we've set up their distribution, it's hopefully going to work. The artificial reference material A covered trisomy 13, 18 and 21 and Aneuploidy. Due to the nature of the samples, they're not fit for purpose for SNP arrays, so we couldn't use any laboratory that was performing SNP arrays. But you can see here that trisomy 21 was picked up, trisomy 18 and 13. The Aneuploidy, this lab, I think, has got a contamination issue that we've gone back and they're looking into that because they've got quite a high pickup rate of trisomy 13s anyway, so I think it's not an EQA issue, it's a lab issue. And the WISE Condor, which is one of the data analysis programs set up in the Netherlands, didn't quite like the artificial material. It wasn't truly reflective of patient material. But saying that, they got the right results. I think it was a caveat because it was EQA. Everybody's a bit worried. When we looked at, so all those results, let's just go back, were at fetal fraction 12%. So you would be expecting everyone to pick them up. The next set of reference samples, B, all were trisomy 21, but we wanted to determine, not test the labs, but determine maybe where the boundaries of limits of detection were. So we used trisomy 21 at 8% fetal fraction, which is here, and everyone detected trisomy 21. 4%, everyone picked it up at 4%, and actually it's nice to see that the iron proton was picking up the percentages at pretty spot on, actually. 2% is where we start seeing a slight difference. Now, bear in mind that these are a small number of labs, so conclusions can't really be drawn, but it does indicate that the Illumina platform doesn't pick up trisomy 21, and here we're also getting trisomy 13, so you wouldn't really report 21 if that was the case. The iron proton picked it up perfectly, as it did at 1% as well, but not with the Illumina platform. So just a snapshot of what we might see with the full EQA. So nearing the end now, stage 4, this is coming up to current. We've actually delivered the EQA pilot for NIPT for aneuploidies. This was delivered at the beginning of 2017 using artificial material. We thought it was better to get something out there just to help labs benchmark their service, even if it was just for a limited number of labs. It was limited, as I say, because of the limitations of the artificial material to just NGS performing labs, and we had 41 labs participating. The assessment is underway now. On Tuesday, we have an assessment meeting to review all the cases and work with the assessors. And we're only going to score the genotyping and just give interpretation comments returned to labs, because I think it's too much to do it in one go. And really, we're just scoping out what should be on the report. But we do know what the genotype should be, and I think that should be scored. Looking at the preliminary data, we do have 3 genotyping errors. Now, in my view, 3 out of 41 labs is not very good. So I think there's a lot of work to be done there. We don't know the cause of the errors. It may be something very simple, like a transcription error into the report, or it might be a fundamental issue within the lab, or a sample swap. And we do see all different types of scenarios popping up. So I guess a real steer that we do need to keep delivering EQA in this field. And we're hoping for the results to be issued mid-June this year. And we've just heard that we have our oral presentation at ISPD this year in San Diego to discuss and present the results of this pilot. And at that meeting as well, we're holding a pre-congress course on EQA for NIPD. So if anyone wants to be involved, then please get in contact with me. We're pushing ahead with Pilot 2 now. We want to be inclusive, so we've taken a slightly different approach. We're sourcing patient plasma, so we can open it out to all methodologies. So we don't want to exclude any labs that are not doing NGS or single paired read NGS. We've got a plea out to all labs who completed the survey who indicated they could provide some patient plasma to us, and we've been in contact with them. So if you've not had an email from any of the EQA providers and would like to participate and would even better be able to provide us with some plasma, then please do get in touch with the NEQAS scheme. So registration will follow the close of Run 1, which will be mid-June, July, and the EQA distribution will go out in autumn. So just finally, acknowledgements to the Specialist Advisory Group. Our validating labs who help out enormously, our peer assessors, who I think do quite like going through all the reports and seeing everything that's going on, and of course the EQA team and the laboratories. Thank you.